all the participants. And now we are about to have the third webinar of the series on emerging and re-emerging viruses, organized by Kerala State Council for Science, Technology and Environment and Institute of Advanced Virology, Thiruvananthapuram. And previously we had two lectures, one by Dr. B. Iqbal, a member of the Kerala State Planning Board, that's on Kerala experience in COVID-19 control. And second, we heard about the biology of SARS-CoV-2 COVID-19 by Dr. Gauri Shankar, who is an Associate Principal Scientific Officer at Merck USA. And now we'll be hearing on to some different aspect. That is the role of non-human primate models in combating emerging viral diseases. And we are indeed privileged to have with us Dr. Koen van Rompay, who is currently full research virologist at California National Primate Research Center, University of California, Davis. And I'll say something about the speaker, that we have circulated a brochure. Along with that, you might have seen a video on his inspirational work in India. And Dr. Van Rompay's expertise is in non-human primate models of HIV infection, Zika virus infection. His research has a special focus on vaccines and antiviral drugs to prevent or treat infection with these viruses, including in animal models of pregnancy and infancy. And he was on the forefront of developing HIV treatments at the California National Primate Research Institute Center in the 1990s, and helped to develop and test the antiviral drug, the tenofovir, which is currently, as part of combination antiretroviral therapy, the most frequently used HIV drug in the world. And tenofovir has saved the lives of many infants by preventing the mother-to-child transmission. Is now the key ingredient in the pre- and post-exposure prophylaxis and regimens to prevent transmission of the virus in high-risk groups. Dr. Koen van Rompay founded the Sahaya International, that's a non-profit organization, a diverse network of friends and research scientists committed to making a difference towards improving the quality of life in third world countries. And through this, they were able to give information or awareness about HIV or AIDS, given access to life-saving HIV treatments to families, hence improving their health and life standards. So with immense pleasure, let me invite Dr. Koen van Rompay for the presentation. Thank you, sir.
All right, thank you all very much. I hope that you can all see my screen. So thank you so much for the invitation. I'm very honored to be presenting to you today. So during my presentation, I'll give a very brief introduction. Then I'll talk a little bit about the role of non-human primate models in biomedical research. And then I'll give some examples of different infectious diseases. So basically, my background is veterinary medicine. I went to vet school in Belgium, and then in 1989, when I got my degree, I was able to get a fellowship, and then I came to UC Davis here in California, and I got involved at the California National Primate Research Center. So our research center is part of a network of 7 primate research centers that are mostly funded by the government. And so of course, we are here in the far west in California. Because of the climate here, we have a very large outdoor colony. So we are actually quite self-sufficient. We have a breeding colony of about 4,500 rhesus monkeys. And so this is the species that's most frequently used in research. And of course, to keep these animals healthy, we have a large staff of primate medicine. They do procedures both, of course, to keep the colony healthy, because they are also very much involved with our research. And of course, also we have a very large staff to take care of the animals. We also want the animals to really be happy and healthy. And of course, here at the center, we have different research units. And so I am part of the infectious disease unit, which is our largest unit here. And so then I'll move on quickly to talk about the role of non-human primate models in biomedical research. So of course, when we talk about biomedical research, it's really important to know and how it's different from the use of animals for example, for human consumption. Biomedical research, by definition, is really we study a living organism with the purpose of healing diseases. And of course, it also means research. We do very careful examination. And of course, it's always good to remind ourselves that the beneficiaries of biomedical research is not just us humans, it's also pets, and biomedical research also makes a very big difference in trying to save endangered species. Of course, whenever we use animals in research, especially when we develop our protocols, we always think of the 3 R's concept. The first question we always ask is there a way where we can replace it? Can we get the same question done by doing in vitro work or by using mathematical or computer modeling? If we feel that we have to use animals, we always try to limit the number of animals to actually address the research question. And of course, also, we always try to refine. We try to, in a way that we can reduce pain and discomfort. Of course, many of you have heard of the pipeline of clinical development for vaccines and drugs. And so basically, the pipeline is basically in several stages. The preclinical pipeline is often where you have a sequential process of screening vaccines or drugs. It usually goes from something in vitro to mice or rats. Then if something still looks promising, then you go into monkeys. And then eventually, if that looks really good, then you go into human clinical trials. And if those human trials go very well, then of course something can get official approval for widespread use. And so this whole pipeline is really a mechanism to sort effective and safe drugs or vaccines from ones that would be not effective or that would be harmful. And of course, it's really important that we also learn what we observe in humans, so that we really provide feedback and use that information to really optimize the in vitro and also the animal models to really make them as relevant as possible. Of course, for most interventions, we always start with in vitro studies, because that really allows us to understand the structure, the replication cycle of a virus. And it also allows us to screen a large number of compounds. But as you all know, in vitro doesn't always mean that something will work in vivo, because there are many compounds that may work very well in the test tube, but that may be too toxic or don't really work in vivo. So that's why, of course, then we often move from in vitro to animal models. And of course, the value of animal models is very high. There are several things that, of course, we can do in animals. We can really control many of the variables. We can really control the species, the age, the sex. We have full control over what do we use to inoculate the animal. Which dose, which strain. Do we want to use a virus that has certain mutations? And of course, we have also very much control over items like timing, which route we give the virus. Of course, we also have full control over the ability to collect samples. We can, for example, collect samples every day or even more frequent. We can take biopsies. We can also euthanize animals to collect tissues. And of course, also third of all, we can also have the ability to do very controlled interventions. Of course, the ideal animal model to study a human infectious disease has several features. Of course, we like the animal model to be as close as possible to the human host. Secondly, ideally, we like to use the same infectious organism as the one that causes disease in humans. And third, ideally, that organism causes a very similar disease. Of course, it's important to realize that there is no animal model that is 100% perfect. So we really do the best that we can. We really try to choose the appropriate animal model to address certain research questions. And so why do we use monkeys? Of course, monkeys are the laboratory animal species. They are the closest to humans. Their immune system physiology are very, very similar. So that's why for studying many human diseases, they are the best animal model. The only drawback is that doing research in monkeys is extremely expensive. So to kind of go back to the pipeline of clinical development, of course, if something looks really good in the preclinical stages, something can actually go to the clinical trials. And so usually there are 3 phases, phase 1, which is usually a very small group of people, which is mostly just to see if something's safe, does it seem to cause the proper response. If that looks promising, then you go to phase 2. That's usually with, let's say, a few hundred people. That can take months to years. Then, of course, you go to phase 3, and that's really where you determine, does your intervention really work? And that can take years and sometimes longer. So if phase 3 looks very promising, then the government can decide to approve the intervention, whether it's a drug or some vaccine, for general use. Then usually after something is brought on the market, there's still what they call phase 4. Because it is always possible that there may be some very rare side effect that was not found during the phase 3 clinical trials. So that's why it's always very important to do follow-up after something has gotten official approval. Of course, as you'll hear later during my talk, sometimes there are situations where it may be difficult to do a phase 3 trial. And in those scenarios, animal models can have even a higher value. Because then sometimes what is possible is that you use data of phase 2 human trials and combine that with efficacy data in an appropriate animal model. And then there may be the possibility to get approval of the drug or the vaccine. So now, of course, I'll jump to some examples of infectious diseases. So, of course, as all of you know, modern medicine over the past 60 or 70 years has seen amazing major victories. For example, there is a very effective vaccine against polio. We have been able to basically eradicate smallpox. There are also very strong antibiotics against many bacterial diseases. But yet, especially during the past decade, it's clear that, as the WHO also announced in a report in 2007, currently infectious diseases are emerging at a rate that history has never seen before. And of course, here I'm kind of pitching to the choir, because as all of you know, there are a number of reasons. We humans, we are really destroying environment. We are traveling a lot more. There is global warming. Most new infectious diseases come from wild animals. So they are what we call natural reservoirs. Often these animals don't have any symptoms. So they just carry the virus and they don't really get sick, but they can be shedding it. And of course, then those viruses can infect humans, sometimes through direct contact. Sometimes it goes through other animals, for example, domestic animals, through vectors such as mosquitoes. And the reason why, especially the last few decades, there is a high increase is because there is so much more chances of transmission. There is a lot of increased contact, either in the wild, in suburban areas, also animal markets. There's also a big increase in the territories of mosquitoes. And also for some viruses, it has been seen that the virus can sometimes mutate to increase the range of hosts and vectors. So how can animal models help? Of course, when there is a new emerging disease, we're basically dealing with this black box, a lot of unknowns. And of course, the first thing we really want to do is get to know our enemy better. And so that's why animal models can be extremely useful to really figure out what are some of the viral and host factors that determine viral pathogenesis. Yeah, what determines whether a host will develop disease? How does the virus transmit? What kind of an immune response does the host develop? Is that immune response protective against re-exposure? Of course, once we kind of understand the animal model, then we can really use that knowledge to study interventions. Whether we're talking about active or passive immunizations, or also about antiviral drugs. So, of course, as was mentioned, I spent most of my career working on HIV and AIDS. And so the non-human primate model has been extremely useful for that. HIV actually comes from monkeys in the wild. Many species of monkeys in Africa are naturally infected with what we call SIV, simian immunodeficiency virus. But when we infect macaques of Asia with SIV, they develop disease just like humans who are infected by HIV. And so we've really been able to use the macaque model to learn a lot about how does the virus cause disease, and also then really to develop drugs to treat infection, and also to prevent HIV transmission. And so, of course, the things that we're still looking for is an HIV vaccine. We've been working on it for almost 40 years. Some vaccines had partial efficacy, but nothing has been found that really has sufficient protection. And of course, we're also still trying to find the HIV cure. Currently, people who are HIV infected have to take medicines daily for the rest of their life. Right now, of course, what's really promising is there is a lot of research going on trying to develop long-acting antiretroviral drugs. And so I'm actually also involved in some of those early stages. And so, like yesterday, I was actually on a call with other researchers, and they think that probably in the next few years they can develop formulations of some of the currently used HIV drugs where you could give a single injection, and that would provide sufficient drug levels for perhaps 6 to 12 months. So that would really give, I think, a major improvement in drug adherence, because then it would be a lot easier for people. They don't have to remember to take their medicines every day. They just go to the hospital every 6 to 12 months. Of course, as you all know, there are many viruses and diseases that are by mosquitoes. So Aedes aegypti and Aedes albopicus are the culprits for several diseases such as dengue, chikungunya, Zika, yellow fever, and other viruses. In 2016, all of a sudden, as you may remember, Zika made the world news. I remember I was in a coffee shop and all of a sudden I saw the headlines, Zika virus. And I have to admit I had never heard of Zika virus, so I simply kind of Googled because I was curious, is anyone working on Zika virus infections in monkeys as an animal model? So I actually found out that the only publications of Zika virus in monkeys actually were almost 70 years ago. Zika virus was discovered in the Zika forest in Uganda. So researchers were actually studying yellow fever. So they put rhesus monkeys in cages high up in the trees. And one day they had a monkey that had some fever. They thought, oh, this is very interesting, let's take a blood sample. And they were able to isolate a new virus from that. So that actually happened in 1947. And because of the location, the Zika forest, they decided to call it Zika virus. So then for many years, there were very little reports about Zika virus. There were some occasional reports that some people got infected, may have had some mild symptoms. But yet, since about 2007, the virus slowly started to spread over the Pacific Ocean. And things really got bad when in 2015 it hit South America. Because there was a very large population that was totally naive. They had no herd immunity. Zika infection of human adults is quite benign. 80% of people have no symptoms, so they don't even know that they got infected. 20% of people have symptoms, but usually those symptoms are quite mild, and they only last about 1 to 2 weeks. So it's mostly some fever, joint pain, rash, conjunctivitis. A very low percentage of people develops what is called Guillain-Barré syndrome. So that seems to be something where your immune system is trying to attack the virus, but there is some cross-reactivity of your immune response against your nerves. But yet, even with the Guillain-Barré syndrome, most people recover within several weeks. So overall, in adults, it's not a very bad disease. But yet, what's really very bad is when women who are pregnant get infected with Zika virus, because there is a high chance that Zika virus crosses the placenta and reaches the fetus. And it causes what we call congenital Zika syndrome. The most prominent feature of that is that there are some babies born with very small brains and very small heads, so what we call microcephaly. It's also been seen that there is an increased rate of fetal death. But even babies who are born with normal head size often have growth restriction, other brain abnormalities, they have eye lesions. And so right now, there is 2020, there is no vaccine or drug against Zika virus on the market yet. What's also been kind of worrisome is that there have been reports out of Brazil that sometimes microcephaly may develop after birth. So for example, here in this report, they had 13 infants that were born. Right after delivery, when they measured the head size, everything looked normal. But then when they followed up those children for 1 to 2 years, they saw that some of them actually developed brain abnormalities. So it's really what we see at birth may just be the tip of the iceberg. Of course, when I then found out that Zika virus can infect monkeys, based on studies in 1947, I was very curious to know what are the studies that we can do right now. And so what was very fortunate is that we formed basically this very large collaboration with other primate centers in the USA. We had weekly conference calls to really share our ideas, share the first experiments that, let's say, that we wanted to do. Because, of course, one of the first questions was, are the Zika virus isolates that are circulating right now, are they still infectious for monkeys? And so we then did those kind of studies in 2 steps. First of all, can it infect non-pregnant monkeys? And if the question was yes, then of course we would move on and then study what's the effect if we use Zika virus infection of pregnant macaques. And so when we infect non-pregnant monkeys, we see, yes, the virus can replicate. And if we monitor virus replication in the blood, then we basically see this kind of a time curve. So on the y-axis, you see the viral copies of RNA per ml of plasma, and on the x-axis, the timeline. And you see that similar to what has been found in humans, if you infect non-pregnant monkeys, you have a short burst of virus replication that usually lasts for about 5 to 7 days. And so the reason it's very short is because the host, humans and also monkeys, develop a very strong immune response. So we basically see within about 1 to 2 weeks the development of neutralizing antibodies. And they seem to then protect, because if we expose monkeys, and it also looks for humans, if they get re-exposed to Zika virus, they seem to be immune. So that's actually very good news. However, what we've seen in pregnant animals, once we started to do those studies, is that we often see prolonged viremia for several months. So there is prolonged detection of virus in the bloodstream. But yet, when we take the plasma of those monkeys and we test for antibodies, we see those monkeys develop also neutralizing antibodies. So what kind of explains why some of those monkeys have prolonged detection of virus? We and also other researchers at other centers have actually seen that sometimes we see fetal death in monkeys. So when we don't see a heartbeat or an ultrasound, we would do a C-section to remove the dead fetus. A few days after we do the C-section, when we take a sample of those animals, we see no plasmaviremia. So this kind of suggests that prolonged detection of virus basically reflects that the virus is replicating in the placenta and the fetus and is spilling over into the bloodstream of the mother. So, as I mentioned, of course, we can infect pregnant monkeys. We see prolonged. So far, nobody has been able to induce the typical gross microcephaly in monkeys yet. But yet, we and also others have seen that we see lesions in the placenta because the virus loves to replicate in the cells of the placenta. We also see increased fetal loss, and we also see brain lesions developing in the fetal monkeys. Of course, we didn't want to use this model to develop an intervention. And of course, the goal is still to develop a Zika vaccine. The goal of a Zika vaccine is mostly aimed towards protecting against congenital Zika syndrome. So what we would like to do is have a vaccine on the market that you could immunize women of reproductive age before they would become pregnant, with the hope that they would develop a good immune response, they would develop high levels of neutralizing antibodies that would then protect them during pregnancy if they would get bitten by mosquitoes. So the vaccine study got involved with is basically a DNA vaccine, which expresses some of the outer proteins of the virus. And so this is a DNA vaccine that the regimen that was developed is one where you would give 2 doses of the vaccine 4 weeks apart. And so it was already shown in 2016 that this vaccine works in non-pregnant adult monkeys. Based on those promising data, they started a phase 1 human trial, of which data were published in 2018. And then that looked really promising because they saw that it gives a very good safety, and it also induces good neutralizing antibodies. So based on those results of phase 1, they actually started a phase 2 human trial in 2016. But in those human trials, they are limiting enrollment to non-pregnant people. So of course, then there is still the question, does this vaccine really protect people? And especially, do we know, or can we find any evidence that it would protect the fetus? And that's, of course, becoming more difficult, because although the peak of the outbreak of Zika virus was in 2016, since then there has been a big drop in Zika cases. So that really makes it a lot more difficult to do a phase 3 clinical trial. For example, this is what has been seen in Latin America. You see that there was a major peak in early 2016. From 2017 onwards, there have been very few cases. But yet, Zika virus is still a big threat. This is an article that was published in the New York Times almost 1 year ago. There are still many places in the world where the mosquito that can spread the virus is present. And even in Brazil, there are areas that have the mosquito, but so far have not had a Zika virus outbreak. As also mentioned, due to global warming, the mosquito vector is also spreading. So there's really a very big concern that it's not a matter of whether there will be a next outbreak. The only question is, when will it happen? Yeah, I actually saw a short report that just even the past month in Brazil there were more than 500 confirmed new Zika cases. Yeah, so it's like right now when herd immunity is going down, there is a very big chance that there will be another outbreak. So the Food and Drug Administration here in the USA, they have what we call the animal rule. So in cases where it's really difficult to do a phase 3 human trial to test efficacy, they can actually grant approval based on well-controlled human studies. And one of the conditions is that you really see very good efficacy in a relevant animal model, and that your correlate of protection is something that you can correlate with an outcome in phase 2, let's say in the phase 2 human trials. So basically that you can see that what gives protection in the animal model is a similar mechanism of action that seems to be induced by the vaccine in humans. So of course, that's where we then got involved. We were asked by the NIH to study this vaccine in a pregnancy model. So of course, the big question is, can this Zika DNA vaccine, if given to monkeys before pregnancy, can that protect the fetus against congenital Zika syndrome? So basically, this is just a very short experimental design. We gave 2 doses of the vaccine. Then we were breeding these female monkeys to get them pregnant. These monkeys became pregnant, ranging from a few days to more than 1 year after they got the DNA vaccine. So once they became pregnant, we exposed them to Zika virus several times. And then, of course, the big question is, does the vaccine reduce transmission? So this is actually the same experimental design, but in some more detail. So here on the top, we have 18 monkeys that we gave the vaccine 4 weeks apart. Then we tried to get these animals pregnant. We got 13 animals pregnant. The gestation of rhesus monkeys is about 165 days. So we decided to expose those animals 3 times to Zika virus on gestation day 30, 60, and 90. So that basically corresponds to to the first and second trimester. In contrast, control animals which did not get a vaccine, and so we also exposed those to virus at the very same time. We sampled those animals many times. We took blood samples. We also collected amniotic fluid. We did ultrasound monitoring, and so then we actually utilized the mothers and also the fetuses at the end of gestation to have access to tissues. Basically, since I don't have time to go over the data in much detail, I'm just going to give a very brief summary. So basically, we saw that the vaccine worked really well. If you look at the clinical outcome, in the control arm, there were actually 2 fetuses that died early. They actually died within 30 days of the first exposure of the mother to Zika virus. In the vaccine arm, we had no early fetal losses. We saw that if we collected blood samples from the mother, that the vaccine, that the animals that got the vaccine had much lower virus levels. Many animals had no detectable virus in their bloodstream. We also saw that there was a correlation between neutralizing antibodies that were induced by the vaccine. the higher the neutralizing antibodies were at the time of the first Zika virus exposure, the lower the chance to find any evidence of virus replication in the blood of the mother. And what was actually also good is, as I mentioned, there were some animals that became pregnant very late after DNA vaccination. So we had some animals that became pregnant 1 year later. And even then, the vaccine was still effective. Of course, the most important question is, did the vaccine protect the fetus? When we looked at the infection rate of the fetus by doing RT-PCR, we saw that in the control arm, 11 of the 12 fetuses had evidence of virus replication. In contrast, 0 out of the 13 vaccine arm fetuses had evidence of virus. And also when we looked at histopathology, that also basically confirmed the same thing, that the animals in the vaccine arm had much less pathology than the animals in the control arm. So this is really very exciting, and if any of you have more questions, if there's time during the Q&A, I'll be happy to talk more about this. Otherwise, we also published these papers, these data end of last year. Of course, the thing is with the vaccine, it always takes a number of weeks before you build up immunity. So we actually did a follow-up study that was published several months ago. Instead of giving a vaccine, what we did here is we used the same experimental design. We had animals that were pregnant, we exposed them to Zika virus 3 times, but then we actually gave them a combination of monoclonal antibodies 1 day before exposure. And we actually saw very similar designs. We basically saw that you reduce the risk of transmission of the virus from mother to fetus. And so this is basically something that would show that if there's ever an outbreak and you are worried that there isn't really enough time to start immunizing pregnant women, this is something you could do. You could immediately give antibodies to pregnant women, while of course at the same time you can give them the active vaccine, so that even during the few weeks that it would take for the active vaccine to induce an immune response against the virus, these antibodies that you give passively will still be able to cover that window and actually protect the mother. Of course, I'll now talk a few minutes about Chikungunya virus, which I know is also very, let's say, relevant for your state. So, of course, as all of you know, this is a virus that was first discovered in 1953 in the Tanganyika region of Tanzania. And so the name Chikungunya actually stems from the local language of Kimakonde there, and it basically means contorted posture. And it really is caused because many people who recover from Chikungunya have chronic joint pain and actually walk in a bent shape. And of course, this is also transmitted by the same mosquito as the one that transmits Zika virus. As all of you know, Chikungunya has really been spreading, especially since about 2005. And so there have been more than 5 million cases worldwide, of which about 2 million in India. And one of the reasons of the very rapid spread is really that the virus developed a mutation in the E1 envelope. It was seen that just with a single mutation, the mutant virus could also replicate in a second vector, Aedes albopictus. And of course, if you increase the number of mosquito species in which the virus can transmit, that of course really promotes the spread of this virus. As all of you know, the symptoms of Chikungunya range, from fever, headaches, but what's quite typical is joint pain and muscle pain. Even although many people recover, there are many people who really have joint pain and muscle pain for months and sometimes years. And so that's really what, the big problem with this. One of the things that's typical also with Chikungunya outbreaks is that they are quite sporadic, so it's very difficult to predict where the next outbreak will be. But once it happens, it's usually quite explosive. And so there's no vaccine or antiviral drug available on the market yet. So other researchers at the National Institute of Health here in the USA, they had developed a vaccine against Chikungunya in 2010. And so they actually published about it. So it's actually a virus-like particle that expresses the outer parts of the virus. And they showed in monkeys that this vaccine protected against virus replication. So of course, this is something that they published 10 years ago. As I mentioned, often vaccine development can be quite slow. They then started a phase 1 human trial. The results of that were published in 2014. So this was basically based on 25 people, they saw that vaccine induces very good immune responses. These data were then used to move this vaccine in phase 2 trial. They published these results about 2 months ago. This was based on 400 people. They also saw that, yes, the vaccine is safe and you get good induction of neutralizing antibodies. So basically, these antibodies are able to inhibit virus replication in vitro. But then, of course, as I mentioned, it's difficult to do a phase 3 human trial because you don't really know where the next outbreak may happen in the world. And so, of course, that's something where then monkeys can actually come in. And so where monkeys can actually then come in is then to try to establish an immune correlate of protection. Basically, if you want to use the FDA rule, where you want to use animal data, see animal efficacy data, to get a vaccine approved for humans, you want to show that there is some kind of an immune correlate that's dose dependent. Yeah? And then you want to show that that immune correlate also is actually consistent with what you see in humans with regard to immune responses induced by the vaccine. And so that's why what we then do in those experiments is we basically do dose response. We give different doses of the vaccine, hoping that we see this kind of a response where with increasing vaccine doses, you see an increased immune response. And at some kind of a threshold, your immune response is so strong that if you infect the animals, you don't see much virus replication and you also protect against disease. And so that's why we then actually work together with one of the biotech companies here in the USA that got a license to try to commercialize this vaccine and try to bring it on the market. So we actually tested different doses of this vaccine. We saw the induction of neutralizing antibodies. The titers that we saw really depended on the dose of the vaccine. And we saw that especially at the higher doses, we saw protection against virus replication and also protection against disease. So we actually saw that control animals would develop joint pathology. But animals that got the vaccine, they were actually protected against joint pathology. And what's also nice is that the amount of antibodies that we saw in the monkeys that you needed to get protection in the monkeys was very similar to the amounts of neutralizing antibodies that are seen in the phase 2 human trials. As I mentioned, in the phase 2 human trials, they saw that these antibodies inhibit the virus in vitro. Of course, if you want to then use this monkey data to try to convince the FDA to approve this vaccine, you really want to build your case, yeah? And for somebody who is skeptical may say, oh yeah, but you show that the vaccine works in monkeys, and you may have antibodies in monkeys, but the vaccine in humans also gives antibodies, but those antibodies may be different. Yeah, they may, even although they neutralize the virus in vitro, that they still act in a different way than the antibodies that you found in monkeys. And so that's of course why we then did, actually a follow-up study to address the question, are vaccine-induced antibodies in humans, can they be effective in vivo? And the way that we then did it is by doing passive immunization studies. So instead of giving the vaccine to monkeys, what was done is in human trials, as I mentioned, they immunized humans. They would collect large amounts of plasma. They would isolate the immunoglobulins, purify them. Then we would give those immunoglobulins to monkeys. And so we would give different doses of those antibodies, and then 24 hours after we gave the antibodies, we would then expose them to the virus. And right now we are analyzing the data, but the data look really good. So we actually saw that there is a dose-dependent protection. The more antibodies you give, the less virus replication, and also you have less pathology, especially of the joints. So this is basically very promising, and so the company that we work with is also really hoping to use this data to go through the whole procedure and hopefully get this vaccine on the market in the next few years. Of course, as you can guess, right now, everyone is always talking about COVID-19, and so we also started to do studies on this in the past few months. So as you all know, there are so many research efforts happening right now on interventions. Right now, there are more than 100 vaccine candidates that are being developed by different laboratories. At least 10 of them are already in clinical trials. With regard to treatments, there is one drug that's currently approved here in the USA to treat people who are infected, which is the antiviral drug remdesivir. And so it has been found that that has a moderate effect and actually by basically improving the time to recovery. But there are also many other compounds that are being tested or that have been tested. Many of them are quite controversial yet because there is often not very strong evidence that they really work. And so some of those, it's really difficult because the only way to really know if an intervention works is by really doing a true placebo-controlled trial. Yeah, but of course takes lots of time, takes lots of money. You need to have a large population of people who are infected. Also in the future, especially once you have some treatments that are found to be effective and are being used in the clinic, it will be much more difficult to test additional interventions. But it will be very difficult to do a true placebo-controlled trial because once you have something that works, in the future it will be likely that your control arm will actually be getting the best available standard of care, which may even then already include, for example, certain antiviral drugs or certain antiviral monoclonal antibodies. That's of course where then animal models can be very useful. We and also other research groups have been developing the non-human primate model, and we see that if you infect macaques with coronavirus, you also get a disease that's very similar to humans. We see clinical signs, we see evidence of virus replication in the respiratory tract. Animals make an immune response. We see lung lesions. When we look at the lungs, we see evidence based on the histology of pockets of pneumonia. So there is basically all the evidence that supports that this is a good animal model. So far, most of the studies that have been done in monkeys are in healthy adult monkeys, and so the disease is similar to what you would see in most healthy adults, where you see like moderate to mild disease. Also, what's promising that in the monkey studies so far is that if you infect monkeys, they develop immunity. So if you expose them to the virus again, about 1 month later, they seem to be immune. They seem to be immune against re-exposure. So that's kind of promising. There have also already been several studies that have been published the past month on testing vaccines and drugs. But yet there are still many questions that aren't really answered yet, and that we hope to do also with monkey studies. For example, can we learn more about what are some of the predisposing factors? And so we're hoping to do monkey studies in the next few months where we use monkeys that are old, monkeys that have, for example, hypertension, type 2 diabetes. Are those monkeys more likely to get severe disease? And what are really the reasons? Of course, we also want to know what's the durability of the immunity. Yeah? Because, of course, ideally, you want to have an immune response also when you vaccinate that would give long-lasting immunity. Yeah? Because it would be too complicated if you have a vaccine that would, for example, only protect for, let's say, several months, and you would actually have to give people a new booster shot every few months. Of course, also one of the big advantages of non-human primate studies is that you can really test interventions and really have a true placebo arm. So that's why we are all now gearing up to do a lot more studies. It's very, I'd say, quite intense. Yeah. One of the things, of course, that's for us also been kind of surprising, as you can imagine. There is such a big urgency to develop some interventions that instead of the clinical pipeline where you first go from in vitro to mice, then to monkeys, and then into human trials, right now everything is kind of like mixed up. Yeah, right now there are a lot of phase 1, phase 2, and soon also phase 3 human trials with vaccines happening, also with like other interventions. Where often we don't even know in monkeys whether that really works. Right now, many of the studies, instead of happening in a sequential manner, are now happening side by side. And so, of course, it has high risks. So it's possible that lots of money and lots of resources may be spent on human trials in testing vaccines or drugs that eventually may not really work. So we will just see how that will kind of evolve over the next year. But of course, we're also really hoping that we can develop a vaccine and get it on the market in the next year. So one of the questions that has also come up with COVID-19 is if it's difficult to do a phase 3 human trial, there has also been more talk about doing controlled human infection. So that basically means that you look for volunteers who agree to get the vaccine or get placebo and then be exposed to COVID-19 virus. Of course, it has a lot of ethical concerns, and you really have to develop a very careful risk-benefit balance, yeah, because the good thing is you will know quite fast whether your vaccine works. But of course, there's also a risk to the volunteer. Yeah, so that's why if you want to do that, you would probably want to have low-risk populations, which means that you want to have young, healthy adults without any predisposing factor. There's, of course, also the talk. Yeah, if you want to minimize the risk that they would even get sick, would it make sense to use, for example, attenuated viral strains? Yeah, so that you have less chance that those people would get sick. But then, of course, it also raises the question: if you find the vaccine that works in human volunteers that are healthy and that works against an attenuated viral strain of COVID-19, can you then translate those positive results to the strains that are really out in the world right now, and especially to high-risk groups? Yeah, so those are lots of questions that will still need a lot of debate, a lot of careful consideration. So before I'll open the floor for questions and answers, I just want to thank the very large research team. This is just a very small fraction of the many people that were involved, especially with our Zika and Chikungunya studies. And so I want to thank all of you for your attention, and I'll be happy to answer any questions. So thank you.
Thank you, Dr. Koen and Rompay. Actually, it was a really interesting and very informative session. And we have a few questions from our participants. So basically, it's around the Zika virus. So does Zika virus produce microencephaly in macaques before or after birth?
So that's a good question. So the microcephaly is often sometimes even seen during late gestation. Yeah, in humans. So sometimes they can even see it when they do ultrasound monitoring of the pregnant women. Some women have decided to actually undergo elective abortion. But of course, as I also mentioned, there are some babies that are born with normal head size and that develop microcephaly later. So far, as I mentioned, in monkeys, nobody has been able to really induce that typical microcephaly. But it's believed that in people, it's only a small percentage of fetuses that may be exposed, may be developing it. The number of monkey studies that have been done so far is still quite limited. I think if I just count all the monkey studies that have been done, all the fetal monkeys, I think we may be less than 100.
Okay. Another part of the question from Ms. Sanjita is, does the virus have specific predilection sites within the brain? That's what her question about.
Yes, so it seems that it seems to go to the frontal cortex, hippocampus. So in the paper that I also cited, we actually have some more detailed data on that, which brain lesion, but it really seems to be that especially also babies with the microcephaly, it really seems to go to the cerebrum. Yeah.
Okay. And another question is about the SARS-CoV.
Yeah.
And in these animal models, does SARS-CoV-2 produce microvascular forces in the lungs, similar to that in humans?
That's a very good question. That's something that we are also planning to look at, and also other groups are going to look at it. So far, nobody has actually described it yet. The thing is, so far in the monkey studies that have been done, we haven't really seen the very severe disease. So far, there are only 3 animals that have been done at other centers that really developed very severe disease and it had to be euthanized. And so the pathologists are still working on doing very detailed histology. So because so far that's something that I think the reason is that few animals have gotten very sick is because, as I mentioned, so far most people have been using healthy monkeys. Yeah, but so that's actually also a very good question. We really want to look at that because there's now a lot of evidence that the virus can really affect the microvasculature, cause blood clotting in the brain, extremities.
Okay. So we have another question regarding the immunity status of selecting the volunteers. So the question is, will it be good to assess the immunity status of these volunteers for vaccine study?
For which virus is it? Coronavirus?
She has mentioned.
Probably, yeah. Yeah, of course. If you want to do vaccine studies, you would actually pre-screen your participants and exclude people who have already seroconverted. The thing is, of course, because we also get that question often, what's the value of testing people to see if they have antibodies against the virus, yeah? Because even here in California, it's almost impossible to get a test done to see if you have antibodies against the virus, even for us as scientists. The thing is, right now, evidence suggests that if you've been infected with coronavirus, you have some immunity against the virus, but we don't really know what's the level of neutralizing antibodies that you need to get protection. Yeah? And so, until we really know that, when people would get tested and they have a positive antibody test, public health officials are kind of worried that people think, I have antibodies, I am immune. Yeah, I don't need to do social distancing. I don't need to use a mask anymore. And so that's why right now they say, even if you have antibodies against the virus, still be very careful. Yeah, because right now we cannot really predict whether you are immune. The only way that we will know that is by really very long epidemiological studies, where we can really follow those people for a long time and get data that way.
Okay, we have another question on Zika virus. So, is the first infection with Zika virus produce enough neutralizing antibodies for the protection in the second delivery?
Yeah, so far, based on the available evidence of animal studies and also in humans, it seems that once you've been infected a first time, you develop enough immunity that lasts a long time, so that if you get exposed months or years later, you may perhaps still get a little bit of virus replication, but since you have immune memory, that immune response would shorten and immediately reduce virus replication. And actually, I mean, anything that you do to reduce virus replication reduces the chances of transmission. So I would say, once you've been immune, once you've been infected with Zika virus once, I think the risk of having complications during a second pregnancy would be very, very small.
Okay. So another question in the same line, does the Zika virus which has infected the baby during the pregnancy period of mother affect the health of the body during the growing periods of the stage?
Yeah, so there is now evidence also coming from many babies, whether they have microcephaly or not, seem to have growth restriction. And so there are some studies coming out of Brazil, and in some ways it can be difficult too, because, for example, we've also been studying it in monkeys, because we actually did some tests where we infected fetal monkeys. We had the mothers deliver the infants, we had the infants grow up, and some of those infants actually, for example, had eye lesions. But those infants, when we test them for Zika virus, we don't see antibodies at all. They lost antibodies, we don't find virus by doing PCR, but yet they have eye lesions, and so it's only because of the history of our experiment that we know that there is a link with Zika virus. Yeah, that's why I predicted in Latin America and also other parts where there is Zika virus, in the next years or next decades, you will probably see many children and youth growing up that may have abnormal brain development or abnormal eyes, but it's difficult to make the link with Zika. And then, of course, it's actually also known from other viral infections, for example, flu. Whenever there is a major flu epidemic that affects pregnant women, flu virus does not cross the placenta, but yet just the fact that the mother has symptoms of flu, she develops cytokines, she develops fever, has an effect on fetal development. And so they've seen that each time when there is an outbreak of flu in the world, babies who are born then, can be totally healthy, but when those children become adults, they have 2 to 3-fold higher chance of developing schizophrenia, bipolar disorder. So also with the Zika virus right now, we don't know what are the long-term consequences. So that's why that's one of the reasons we want to get the vaccine on the market as soon as possible.
Another one is regarding COVID-19. It's does the SARS-CoV-2 produce any neurological symptoms in these animal models?
That's a good question. So far, we haven't seen that in a limited number of animals. We completed our first animal study 10 days ago, so we also collected brain tissue, and we're also planning to look at that. But so far, it looks that the virus is not really neurotropic. The evidence of people suggests that people who develop symptoms of brain disorder is probably really because the virus causes blood clotting, yeah, and causes kind of like small brain infarcts. Yeah.
I think it's the same, a similar question regarding the COVID-19. Like, is it actually dangerous for those who are already having heart and lung diseases? So does it have a great connection with the immune power of or immune system of the person?
Yeah, so it seems that people who have predisposing factors, whether it's heart disease or also lung disease, are at an increased risk. So it really also depends, because, for example, I think I've seen some reports where they said that people who have asthma don't seem to be more likely to develop severe disease, but people who have hypertension, also people, say people who, for example, smoke a lot, have a much higher chance. What's really interesting with this coronavirus is that it doesn't seem to be the virus application itself that kills the patient. It's really the immune response. In some people, it's the immune response overreacts in trying to fight off the virus. The immune response, the immune system in the lungs secretes cytokines, the chemokines that attract more cells. Those cells start to attack lung tissue. They start to attack virus-infected cells. They secrete more cytokines and chemokines, which just does more and more damage. So it's really what we call a cytokine storm. And that's really then what causes the major lung pathology and causes lung edema, and people actually suffocate in their own lungs. And right now, we don't really know why some people develop that, while others don't. Also, available evidence suggests that in many people, virus application stays limited to the upper respiratory tract, like the nose and the throat. Yeah, and so if it's confined there, of course, you have running nose, you have a sore throat for a few days. That's not too bad, but the deeper the virus can go into the lungs, the more and more risk you have.
Okay. So another question regarding coronavirus, that does the coronavirus have effect on the gestation period, or will it lead to a congenital malformation?
That's a good question. So far, there is not any evidence. I reviewed a review paper last week where they summarized all the studies that they could find that have been published, and so far it looks that there is no real effect on the fetus, also even on newborns, because there are some cases, for example, where the pregnant woman herself was not infected, but the newborn became infected, because after delivery, all the family wants to come to visit the baby, and everyone wants to kiss the baby. And so there are cases where the babies actually became infected, but those babies, they had virus in the nasal swabs and the throat swabs, but they didn't really get sick. The biggest concern is actually in pregnancy, of course, if a pregnant woman gets infected later in gestation, and if she has some predisposing factor, for example, some other lung disease, then of course, if she gets compromised with her lung function, because of course, if you have so much pressure of the developing fetus in the womb, that women have a hard time breathing at the end of gestation, that of course can actually be dangerous for the woman. But so far, if you have a normal, healthy pregnancy, there doesn't seem to be any real complications. It's really only, I think, towards the end of gestation, if you basically have a lot of compromising factors at the same frequency, because in healthy, in normal adults, it's about 1 to 2% of cases that go really bad. So far, it seems that also in pregnant women, it's about the same percentage. So just the fact of pregnancy itself doesn't do much, unless if you're really late in gestation and you are in that 1 or 2%, then of course getting the pregnancy may make it more difficult.
Okay. So another, I think the question of what are the chances of getting a second infection after one getting infected and recovered out of COVID-19?
That's actually a very good question. So, based on the monkey studies, it seems that there is some immunity, but of course the monkey studies have been limited. They would expose monkeys the first time, then they wait like about 4 to 5 weeks and then expose them again. Yeah? So which means that you really expose them when they still have quite good antibody response. Yeah? They've also done some vaccine studies in monkeys, but they also challenged the animals quite fast after the last vaccine dose. Yeah? Based on other coronaviruses and other respiratory viruses, there is kind of concern that the immunity may not be lasting very long. Yeah? In the monkey studies that I was just talking about, what they saw is that when you get exposed a second time, you seem to have more protection in the lungs than in the upper respiratory tract, than in the nose and the throat. So that means that you seem to be protected against the disease that could kill you, but you could still perhaps get infected and the virus can still replicate in your nose and throat. You may still be able to transmit the virus to others. Even when you think, oh, I am immune, you may not get sick. So that's why right now my guidelines are, even if you've been infected once, protect yourself against re-exposure, not just to protect yourself, but also to reduce the chances that you would transmit it to other people.
Okay. And another question in the same line, like the effectiveness of this anti-malarial drug, hydroxyquinone, how effective is it against COVID-19?
Yeah, of course, it's still a very controversial thing because almost every day there are more stories coming out whether there is some evidence that it works, that it doesn't work. I think today I saw a short editorial in Science magazine that said that some other studies are coming out that show that it doesn't really work. Yeah? So far, I think if I have to look at all the available evidence, there's more evidence that it doesn't work than there's evidence that it does work. But of course, I know it's, especially here in the US, it has become very political, because our president claims that he's taking it. And so, supposedly, even to do human trials here in the US has become very difficult, because if you ask a volunteer, do you want to enroll in this trial, you may get hydroxychloroquine or you may get placebo. People who are Democrats say, oh, I don't want placebo, I don't even want that drug. But people who are Republican, who support Trump say, oh, I don't want placebo, I want the malaria drug. So it's, my prediction is we really need, I think based on the studies that I think are coming out in the next few weeks, where I think there are also some studies where they really use placebo-controlled trials. Based on the preliminary data so far, there is no evidence that it really works. Which for me is really a big surprise, because you often have things that may work in vitro, but that in vivo totally fail. I mean, I've also done monkey studies with different drugs against HIV, that in vitro it works really well, and then you give it to monkeys, and it actually works very poorly. Or actually, sometimes it didn't even work. That's why I think there is more hope on the use of convalescent plasma. If you can have neutralizing antibodies that you take from somebody who has recovered, and you put those into a patient, there's more and more evidence that suggests that yes, those antibodies can have a therapeutic effect. And right now we actually want to go a step further. I'm actually working with people at the Rockefeller Institute. They have been able to isolate monoclonal antibodies that are very potent. And so with monoclonal antibodies, you can give a much higher dose. Yeah?
Okay. Another question related to coronavirus is that how, can coronavirus disease be caught from a person who is asymptomatic? Because I think the number of asymptomatic cases are increasing. So
that's like a very good question. Because of course the WHO came out with this report. It was like 1 or 2 days ago where they say, oh, it's very rare that people who are asymptomatic. But it's still kind of like the definition of asymptomatic and pre-symptomatic is different. Many people think, oh, I don't have symptoms, but then a few days later they still have some runny nose or a little bit of a sore throat. Yeah. So that's why, since I think right now we don't really know for sure. That's why I think don't assume because you feel healthy that you may not have it. I mean, it's possible that you really may be asymptomatic. Asymptomatic means that you never develop symptoms, yeah? But you may be in the pre-symptomatic stage. That's why we also here tell everyone, just assume you may be in the pre-symptomatic stage. So even when you think you're healthy, wear a mouth mask to protect people around you.
Okay. So we have another question related to coronavirus. That what are the typical clinical signs shown by these non-human primates when they are infected with COVID-19?
Yes, so we inspected them. Most of the animals that we've had some animals that had like some sneezing, coughing. We haven't seen much fever. Yeah, so of course I think it's also similar to most other humans. They have very mild symptoms. It's like some running nose, say a sore throat, perhaps some mild fever. So I'm sure that when we do more, let's say large animal numbers, and especially when we start to study monkeys that have predisposing things, I predict that we will see more severe disease.
In between, there was a few news regarding the effectiveness of BCG vaccine against
yeah. So that's actually a very interesting thing. So I think it's possible that has some effect, but I also want to see more detailed studies. Because basically what BCG does is it actually changes the status of the immune system. It uses what they call trained immunity. And that kind of affects how your immune system reacts also against other viruses. And so it's possible that it may have some benefit, but I would like to see some more data on it and much better understanding. That's I think also why I've been talking to some of my also some administrative in monkeys, because we could immunize some monkeys with BCG and see how does that affect coronavirus replication.
So I think we have a lot of questions, but I'll just tell you the final question and we will wind the session. So it's regarding herd immunity, like especially in countries like India, we are hoping to achieve this herd immunity as far as possible. And what's your opinion in that regard?
Yeah, of course. That's kind of what we all hope for, that there eventually will be herd immunity. Of course, it's to have sufficient herd immunity, you need to have a very large population, say a very large fraction of the population that is immune to it. Yeah. And so, of course, that will take time, and to reach that level, of course, you will have many people who get infected, and many people who also will die. So that's kind of. That's why, of course, what we're hoping to do is, if you can slow the spread enough that hopefully many people can get the vaccine, that we can hopefully get the herd immunity not by transfer infection, but by the vaccine. And that's why right now there's such a big push in vaccine development. There are so many vaccines being developed all at the same time. And I think I see benefit in that, because it's like, even for example, if 10 vaccines work, give some positive effect, all those companies, if they can all pump out vaccines, they can hopefully achieve large herd immunity. Then, of course, for the long future, I see it will be really a question of which vaccine is the most durable immunity. And some vaccines perhaps make immunity for perhaps a few months to a few years, but then we may see that certain vaccine manufacturers may give vaccines that may actually give good immunity for perhaps decades, and those other vaccines that in the long run will kind of take over.
Okay. So I think we have a number of questions from participants. I'm really sorry that I cannot take all your questions because of the time constraints. And I would like to, on behalf of KSCSTE, I would like to thank Dr. Koen von Rompay for this wonderful session. And thank you very much. Thank you to all the participants who have joined the session.
Thank you very much. Thank you very much for your good questions. If you have any questions also that you would like to ask me, feel free to contact me.
Okay.
Thank you very much.
Thank you.
Have a nice day. Bye.
Thank you all participants. Bye.